Hello. All right. Welcome to the last screencast for chapter 15. All right. So I'm trying to make this, oops, sorry. Yeah, froze. I'm trying to make the screencast shorter. So they're each like 15 minutes, which if you watch them double speed and then watch them again, they're shorter than before. So that's why there's a little more of them. Trying that out. I would love some feedback on that. Okay, so let's share my screen and we'll get on to the last screencast for this section of genetics. Is it going? Oh yeah, okay, there it goes. All right, so we talked about genetic linkage, we talked about X-linked chromosomes, and now we're talking about something that can happen in meiosis, which is called nondisjunction. And nondisjunction is just a fancy word for saying bad separation of chromosomes, right? Bad separation of chromosomes. So we know that pairs are really important in genetics, right? And if you lose a pair, then you are in trouble. If you gain an extra pair, you're also in trouble, right? So we are diploid and we want to maintain 2 copies of each chromosome. However, if you have bad separation of chromosomes, you can result in what is called aneuploidy. This is having abnormal numbers of chromosomes, either too few or too many, right? So if we look here at our normal situation, we all have the correct numbers of chromosomes. Otherwise, we would probably have some issues. So we have a total of 23 pairs of chromosomes for a total diploid number of 46 chromosomes. However, if you have aneuploidy, you can have a situation where you result in missing chromosomes or where you have an extra chromosome, right? So monosomy, missing one, right? You have something that has a single chromosome, no pair. Trisomy is where you have 3 of a chromosome. So non-disjunction, or in other words, bad separation of chromosomes, can happen in meiosis, and that can result in aneuploidy, or bad numbers of chromosomes, right? So it can happen during either phase of anaphase, when these chromosomes are separating. If you get non-disjunction in anaphase 1, that can result in having every single gamete having issues, right? You either get gametes that have extra chromosomes or gametes that have less chromosomes. If you have non-disjunction in anaphase 2, then you will result in 2 normal chromosomes, or 2 normal gametes and 2 gametes that are bad, either too many or too few, right? And I really suggest that we try drawing this out. So if we start with too big and too small, so for example, we're going to start with a chromosome, a cell that is heterozygous for both. All right. First thing first, we always are going to remember to duplicate those chromosomes. Right? And then remember, they are going to line up at the metaphase plate, the homologous chromosomes. So I'm just going to say the metaphase plate is here because they're kind of already lined up. And our mitotic spindle is going to be there. So if we have non-disjunction in meiosis 1, you are going to get a situation where instead of having the chromosomes segregate properly, one of them's kind of going to get sticky and they're going to stick together. So let's say these A's stuck together. So what would happen is that the B's would separate okay. You'd have homologous chromosomes going to two different cells. However, then you would have the A's both going to one cell. So that when you, for example, then went through anaphase 2, so here's our metaphase plate, you would end up with a cell that has two A's and a B, two A's and a B, and two cells that had just a B, right? None of these gametes are correct. Remember that gametes have to have one copy of all the genes. So this is really bad when non-disjunction happens in meiosis 1. If you then draw this out again and you look, so let's go ahead, I'm going to go ahead and do it. So let's go ahead and draw out what it would look like if we had this in metaphase or meiosis 2. So we're going to duplicate our chromosomes again to make our kind of X's, our sister chromatids. And we have our metaphase plate. And if meiosis 1 goes okay, we are going to result in a cell that has a duplicated A and a duplicated B for both cells. Then, when we separate our sister chromatids in meiosis 2, we are going to see non-disjunction in one of them. So again, let's say down here, we're going to have this one be sticky. So this is going to be our non-disjunction event. So A is going to segregate properly. So we're going to get one in each cell. But B now doesn't segregate properly. And we have one cell that gets both B's and one cell that gets none. Both of these gametes are very bad. In the other cell, however, no non-disjunction occurred, and you get one copy of each chromosome, right? So non-disjunction can create aneuploidy, or improper numbers of chromosomes, and it's much worse when it occurs in anaphase 1, meiosis 1. Okay, so non-disjunction in meiosis results in aneuploidy because these gametes, those aneuploid gametes that we made, have improper numbers of chromosomes. So for example, if an aneuploid sperm got together with a normal egg, you are going to get an aneuploid zygote, all right? And so our most common example of non-disjunction resulting in aneuploidy is one that you guys are probably familiar with, trisomy 21, which is otherwise called Down syndrome, right? And one in about every 800 newborns is born with Down syndrome, and this goes up with maternal age. And the reason that's important is that as maternal age increases, as mom age increases, you are going to get more likely to have non-disjunction. Okay? And that's because the eggs are sitting around for many, many years, and those chromosomes can become sticky and you won't get proper segregation. All right, so I want to mention very briefly, I'm not going to talk about this very much at all, but I want to mention it in case you read about it somewhere. So females, because we have two X's, we are fundamentally different in terms of the numbers of big chromosomes that we have than males. So most chromosomes, if you have an extra chromosome or a missing chromosome, you're lethal. Down syndrome is a unique situation where you can have a viable person, even though you have an extra chromosome, but that's rare, right? However, females have 2 X chromosomes and males just have 1. That's fundamentally unequal. So what happens is in females is really cool, is that you get X inactivation. And I'm not going to say a lot about this other than one of the X chromosome kind of crumples down on itself and becomes what's called a bar body. And you don't get any gene expression from that. And one of the things that's really cool about this is how you result in calico cats. It has to do with random X inactivation and the pigment colors on that X chromosome, the genes responsible for pigment color, right? So you can read more about that, but calico cats should all be female because of this X inactivation. All right, the last part of this is called chromosomal rearrangements. So even if you have the proper number of chromosomes, those 23 pairs, you can have some funky things that can happen with chromosomal rearrangements. For example, you can have pieces of the chromosome that go completely missing. You can have pieces that get duplicated. You can have pieces that flip, right? And you can also have what's called a translocation, where a piece of a chromosome can hop onto another chromosome, right? And I don't know if you guys know, so 2 examples of chromosomal rearrangements in the real world. So first off, my sister is a genetic counselor. She did her karyotype in graduate school, and she found that she had a translocation between chromosome 16 and chromosome 2 or something. And it didn't cause any issues because all the chromosomes were there, all the genes were there, and luckily that like cut place where those chromosomes got kind of cut and flipped over wasn't like in the middle of a gene, right? Another example of translocation is in the case of Miss Zadalis. So some of you know Miss Zadalis. She has a Down syndrome son. I have asked her if it's okay to share this story, and she's fine with it. Her son, Eric, who's on campus a lot, he's awesome, has Down syndrome. And his Down syndrome is caused by a translocation. He had an extra chromosome 21 stuck onto another one of his chromosomes. So he didn't have a full 3 separate chromosome 21s. He had a piece of chromosome 21, an extra one stuck on another chromosome. Pretty interesting. And she has a karyotype to show that. It's really neat. Okay, so again, lots of practice questions. This section absolutely requires you to do a lot of practice questions. You can think you understand it, but unless you do the problems, you might not get there. All right. Chip, chip, cheerio.